7:15, so we're gonna go ahead and get started. I'm gonna turn it over to Dara. Thank you.
Thank you all for coming again in person and online. So today we have we have Dr. Martin Hamilton. He is a native of Kentucky and has worked and studied at institutions in the USA and abroad, including Bernheim Arboretum and Research Forest, the Royal Botanic Gardens, Kew, and Oak Spring Garden Foundation. Martin is an honorary research associate at the Royal Botanic Gardens, Kew, and a fellow of the Linnean Society of London. He is particularly interested in the conservation and management of threatened species, habitat restoration, and the development and management of plant collections. He is actively involved in international projects that research the conservation and management of threatened species, including the development of ex situ plant collections and horticultural protocols and biogeographic, population genetic, and phylogenomic studies. Martin started as the executive director of the South Carolina Botanical Gardens at Clemson University in August of 2023. Well, join me in welcoming.
Thank you, and good morning. I really appreciate everyone's interest coming to hear about some of my research. As you'll see as we go through the talk, I'm going to start off just a little bit about me because I'm new here. Get to know me a little bit, talk a little more about plant conservation and the need for that. And then we'll pull on some examples of work from my personal PhD project and some subsequent work, mainly while I was based at Royal Botanic Gardens, Kew. I think one of the important things here that I'll flag up is that collaboration is the key to these projects being successful. And hopefully that comes across during the talk. But you know, if you take one thing away, you've got to work with people and you've got to get to know them, build those relationships, and then you can be successful. So I'm a botanical horticulturist. I'm a conservationist at heart and a woody plantsman. So I have a broad background. I've got a lot of experience working both with temperate and tropical plants, glasshouse and field nursery production, and indoor and outdoor public display. I've had a lot of experience working on small islands throughout the world, particularly in the Caribbean and the South Atlantic. And they're a great place to study how ecosystems work and how threat and invasion processes drive speciation and extinction events. And again, you'll see some of those examples as we talk today. I have a really deep interest in the art and the science behind horticulture and trying to convey that to people. The importance of understanding how plants grow from the in and the out is all what horticulture is about. And I've spent most of my career trying to apply the things that I've learned, the experience that I've had, one, to plant conservation and trying to keep species from going extinct, but more importantly, how to communicate that to people so that they understand the importance of our work together to try to overcome the challenges of today, particularly the biodiversity crisis. So why is plant conservation important? Globally, around 40% of our plant species are threatened with extinction. And the risk of extinction for flowering plants goes up to around 45%. In the most recent State of the World's Plants report, which these figures come from, we can see that as we learn more about our environment around us, and particularly the plant species that make those up, we're finding that our actions are driving species to extinction, right to the edge of the point that we will lose them from our ecosystems. And so slowing those trends and hopefully reversing them is really paramount for us to ensure that we don't suffer the most important consequences of the biodiversity crisis. And that ties into climate change. And you'll see some examples of how my research has been looking at that as well. So here in the U.S., at the national level, the U.S. Fish and Wildlife Service takes the lead on recovering, conserving, regulating, and managing our freshwater and terrestrial organisms. And these are listed under the Endangered Species Act. One of the important things to know here is, the ESA is a very comprehensive attempt to provide legal protection to identified species and to consider habitat protection for them, because that's a really integral part. But one of the problems, and particularly for plants, is that the ESA often doesn't have the mechanism to protect species, particularly on private lands. And really can only come into effect on public lands when there are federal funds that have an impact on that. And so this is really important to know. There's a distinction between plants and animals that are listed under the ESA. And you need to keep that in consideration as we're talking through some of these scenarios. Plants can either be listed as endangered or threatened, and those come down to the risk assessments that were done. And we'll see examples from 2 different species that are at different risk levels. So the Center for Plant Conservation helps coordinate our conservation actions across the U.S. And in particular, they register collections that are held by institutions so that it's easier to find information out about them, where those collections are held, who's looking after them, what genetic diversity they hold, and if there's any work that's been done on creating horticultural protocols or gene banking. One of the important things is the CPC also provides links and information to help you understand how best to save plants. So there are best practices, measures, workshops that they host that help people understand what are the most modern techniques we can be using and how do we go about plant conservation in the modern context. So here at the state level in South Carolina, we have around 2,800 native vascular plant species. 333 of these are considered to be priority plants, so ones that we should be focusing efforts on because of some risk of extinction. And that's in our state wildlife action plan. And amongst those, the U.S. Fish and Wildlife has 23 species that are federally listed under the ESA. 19 are recorded as at-risk species. So before a species is actually put on the ESA, they can be placed on an at-risk list, which pushes them up the priority chain for further research to understand if they need to be assessed and listed under the ESA. The state provides us with some really great resources. You can go online, you can find out information at the county level about species distributions. And that links through to the NatureServe website, which is where most of the data is hosted about the species that we're concerned about across the U.S. There's an example here you can see with Pickens County highlighted, and the NatureServe website showing one of our endangered species, the mountain sweet pitcher plant, Sarracenia jonesii. So this is just an example of the sort of information that you already have access to online. And the reason I highlight this is that now the South Carolina Botanical Garden, we really have an opportunity to use these data to generate a priority list of species that we can have an impact on. And this can be through a range of different measures, from seed banking to having conservation collections at the garden, working in situ, and developing partnerships across the state that hopefully will lead to in bolstering those wild populations. So actually growing material out to the garden, reintroducing it into the wild, or putting into suitable habitat in protected areas, or on private lands where we have agreements in place. But I think one of the more important things for the garden is having this material on display so that we can educate the public and really raise awareness about the significance of these plants and how important they are to our ecosystems. So Botanic Gardens Conservation International led a gap analysis of plant conservation concern for the Southeast U.S. And they identified priority species that we need to secure in conservation collections. And using the data from this report and our state and federally accessible information, we'll generate a priority list of species to focus on at South Carolina Botanical Garden. And we're working really closely with the state, particularly the state botanist, to work through that list and figure out which are the highest priority plants that no one's currently focused on that we can help with. Internally, our collaboration across the university is important. One of the reasons I'm here today is to build some links And I think we've got great opportunities across the university because as you'll see, as we kind of talk through these techniques, it's going to take a lot of people that have a lot of different specialisms to understand how best to conserve the species that we're interested in. Externally, building up collaborations and maintaining really strong partnerships with state and federal agencies and NGOs in particular is going to be crucial. And we need to build up our connections with existing regional initiatives. I think one of the most important is the Southeastern Plant Conservation Alliance. So this group is organized by the founding members, which are Atlanta Botanical Garden, BGCI, and NatureServe. And this alliance covers 17 states and territories. I first got involved with this group through my work in Puerto Rico, which you'll hear more about. But they have some fantastic resources. They organize workshops, conferences, and they're helping to prioritize species conservation efforts. South Carolina Botanical Garden can also play a much more significant role in conserving our plants here in South Carolina by following what I think is a really well laid out and thought out plant conservation model. So this integrated model was first put forward in publication back in 2004. And this is something that we adopted at CUE with a lot of our project work. Was trying to follow this flow model through and understand, which pieces of this do we have the ability to do in-house, and which pieces of this do we need to find partners to work with us on. And it's, I think, a really easy way to kind of visualize, where strengths are within your own group and where you need that external support. And in order to do this, we kind of have to understand first some fundamental questions. So what do we first need to think about? Well, what is the species we're trying to conserve? And you may think, well, this is obvious, but it's not. There are examples of species listed under the ESA that we later find out through genetic work aren't what we call them. So figuring out what is that species concept that you're talking about is one of the most fundamental parts of this process. And that can be through literature research, going and looking at herbarium specimens, that baseline kind of taxonomic review of the plants that you're worried about. We have to know publication dates of names and what's the current accepted name for that species. Once we pull together that kind of literature information, we then have to think about looking at genetic studies and understanding how the species fits in the tree of life. What are its closest relatives? And understanding when we go out and we're looking for this thing, that we all have the same idea of that species concept and the nearest relatives, because that also can help inform quite a bit. Oftentimes related species, there is published information about them, but maybe not your target species. So you can use that to inform your process for working through this model. We then need to think very seriously about what genetic diversity is out there. And again, this we can use very modern techniques, looking at population diversity, trying to understand if we can detect subpopulations and populations across the species range, and looking to see if there's any issues with inbreeding, bottlenecking that are going to impact us down the road. And then finally, in this slide, we need to think about the environmental niche that the species fits in. So what are the abiotic things that the species needs to survive in its habitat? And a lot of this we'll pull in GIS data. We'll use that data to look at, does the species need serpentine soils? Does it only grow on limestone geology? And then are there things that we can do in the field through survey and monitoring to build up a much better data set to inform our understanding of the climatic conditions? And we can use remote sensing technologies as well to help us understand these environmental needs. If it's unknown or uncertain, we need to determine the species' reproductive biology. And for a lot of plants, it is unknown. This can be done through field-based work, lab-based work. We need to know, is the plant reliant on some animal for its pollination or for seed dispersal, for example? Are there impediments to its reproduction? So has that animal gone extinct, or is it no longer found in that environment? Is that why we're not seeing dispersal of seeds? We also need to consider conservation measures. So what's appropriate for this species? And more importantly, what's feasible, given the resources we have, the knowledge we have about it. So can the species be put in a seed bank? Can the seeds be dried? If it's an acorn from an oak, no. So we have to look at other measures that we can employ. Can we get it growing in ex situ collections? Have it in botanic gardens and arboretum, share material around so that we conserve genetic diversity that way. Or if we can't do either of those things, then we have to focus on the in situ, and making sure that the species habitat. And that's probably in most instances the more important thing. If we can keep species in the wild where they should be, it's a lot cheaper. It's a lot easier. But oftentimes that's very difficult to do. We also then take all this information that we've gathered and we begin to think about what is the risk of the species in the wild. And there's several different criteria that you can use kind of internationally. There's the IUCN Red List criteria. At the national level, we follow the NatureServe model. And then the federal government for the Endangered Species Act have their own assessment process. And they rely on the information that's available from both Red List and NatureServe risk assessments. So with this in mind, I'd now like to show you some examples from some of my field projects. So my PhD explored the phylogenetic placement, biogeography, and conservation genetics of a threatened shrub that's endemic to the Caribbean. And it demonstrated the importance of designing conservation measures that include potential climate change scenarios, future land use change, and developing representative ex situ collections. I've continued working with this species since I finished my PhD work, and we've used this as a model to develop and work on other projects and other species, particularly in the Caribbean. So just a little bit to kind of set the scene so you know what we're talking about. The insular Caribbean is a biodiversity hotspot. There's over 10,000 indigenous taxa to the region, and around 71% of those are endemic to the archipelago or part of it. So really high endemism, one of those things that really drove the assessment as a biodiversity hotspot. The Puerto Rican Bank is a biogeographical unit, and it comprises 3 different countries. So there's Puerto Rico and the U.S. Virgin Islands, which are both territories of the U.S. And then on the eastern side, the British Virgin Islands, which are one of the UK overseas territories. So the 3 political units, separate funding streams, have often meant that survey work, conservation measures, information about the species hasn't been very easily shared across those boundaries. And that was one of the real driving factors for me to consider this species for my research. So within the bank, there are over 2,000 indigenous taxa seed plants and around 14% endemism. Amongst those endemics is this plant, Veronia rupicola. It's in the Boraginaceae family. So oftentimes in the temperate regions, we know borages as little herbaceous things. Well, in the tropics, they turn into shrubs and big trees. It has a really restricted range. It only occurs in a few small areas, which I'll show you a map of in a moment. And before my project, there was really little known about this species, particularly across its range. There were historic observations, a few specimens, but what was lacking was a real detailed knowledge of this species so that we could move forward with a conservation project. So the genus is a Neotropical New World. There's around 100 species. In the Caribbean, there are 67 species recorded. And the three that are listed here, from left to right, Veronia rupicola, bahamensis, and Lima, have often been confused in the field and reported with overlapping distributions in the past. So again, this goes back to that fundamental research. What are you talking about? What species is it? Going back and checking those herbarium specimens, making sure the identifications are correct on them. So after digitizing all the material held at Kew, I reached out to and visited many herbaria in Europe and the US, pulled all that information into a database, and was then able to begin to build on that with my field records. So as I mentioned, the Puerto Rican Bank is a biogeographical unit. The reason for that is that during the last glacial maximum, that blue outline, it was all one island. So not that long ago in history, this was just one big island. And so you need to understand that to be able to understand kind of the historical context of these plant species distributions. So the bank itself is currently known to have 9 Veronia species left in it. So Veronia rupicola is the endemic to the entire bank. And then there's 2 further species that are endemic just to Puerto Rico, Veronia bolonis and wagnerorum. And you'll see a little more about Veronia bolonis later. So as I mentioned, Veronia rupicola, my target species, has this really restricted distribution. So these little isolated areas on the south coast of Puerto Rico, the island of Vieques, and then across the island of Anegada in the British Virgin Islands. When I first started, I was able to pull together, and I only found that there had ever been like 40 collections made of the species since it was described in the late 1800s. Most of them had little or no locality information on them. You're lucky if they just had the island name, and really nothing about its habitat, other than we know from the description of the species name, rupicola means on rocks. So obviously that's quite helpful that when it was described, Urban told us they found it growing on rocks. So what I wanted to look at using that information was what we could figure out historically where the species have been collected. So this lack of information really drove me to establish a network of collaborators, people I could work with. And I worked on this for about 2 years before I actually started the project. So this was getting permits. This was making the right contacts in each of the countries. Don't underestimate how long these things take. And it's probably the most important thing that you do is building those networks and those relationships with people if you want to be successful in a project like this. Not all of them take this long, but I was working in 3 different countries. So that really added to the issue. I was in the UK and working into international countries. So the network itself, as you can see, made up of many different organizations in country and abroad. And what we started off with was developing an ex situ collection back at Kew, which I'll show you some examples of. So we started off the process by taking seed material that had been banked at the Millennium Seed Bank. We used this to develop a horticultural protocol. So we first did seed germination studies, established those, grew those out, and then we started taking asexual cuttings and learned how to propagate the species. So we could do it both asexually and sexually in controlled conditions. That's a huge thing. A lot of plants you can't do both of those. So this meant that fundamentally any plant in the wild we could replicate. I worked with partners in the British Virgin Islands at their botanic garden, the J.R. O'Neill on the island of Tortola, to establish an ex situ collection. We used a range of techniques, took the protocol that we had written up, and were able to replicate it in local climatic conditions. So again, we had established that this works. We can replicate it. Plants on display then got labeled. They were used for education and outreach purposes. Because one of the big things is people understanding what's in my backyard that I see every day. What's the importance of it? It's not something just to chop down and bring in a foreign plant to plant in this place that looks pretty. So that in itself, I think, is something we really have to keep in mind when we're thinking about plant conservation, is the education side of it. We don't work in a bubble. We have to get that information out. My colleagues in Puerto Rico also set up ex situ collections at some of the state forests, some of the wildlife reserves, and there were ex situ collections already held at Fairchild Tropical Botanic Garden in Miami that I was able to access. We then set to work establishing a new research collection at the University of Puerto Rico Mayagüez campus. And that became then the basis for much of the research that we did at the local level in working with students to go through the process of understanding the species reproductive biology through camera trapping. And also, once again, we demonstrated that we could replicate the horticultural protocol method to get the species into cultivation using new material from the wild. So field work was undertaken across all 3 islands where the species had been recorded in the past. We were able to find extant populations on all 3 islands, documented several hundred new individuals, new subpopulations. So really brought a lot of new information to the forefront. The purple squares that you see here, these are the DNA sampling locations. So all of these samples were included in the genetic analysis that we undertook. And in order to enable this field work, all the data were pulled into a GIS. And this was available to me on a handheld device out in the field running Esri ArcPad software. So the idea behind this is it's digitally born data, which makes it easier to process. It cuts down the risk for introducing spelling errors. The number of times that I've geo-referenced a specimen using what was written on the label and it comes up in the middle of the water. So you get away from all those issues, most of those issues. I won't say all. Desk-based studies were undertaken. So we pulled together all of the information that we could find into a GIS. And so this included geology for the islands, Landsat data, classified habitats, roads, developments, anything that we could find was pulled together. And we used these to understand if the species had associations with specific habitat types, geology types, and how much of that habitat was left. So with the GIS developed, undertook a survey, did random sampling, and the species was only found at 9% of the sampling locations. So that's the first kind of red flag that, we're in the right areas, we're in where it was sampled before, but through random sampling, you're getting very low occurrence data. So using those data and the ex situ collections, I then started undertaking the lab-based work. So DNA samples were extracted from historic herbarium specimens, more modern specimens, and all the field samples. And I also took samples from all the known ex situ collections. And to look at its phylogenetic placement in the tree of life, I selected 2 areas to look at. These were ITS and the Turnell-Turnef regions. So the nuclear region, ITS region, was very informative. We were able to use that and to show us between it and a combined data set with the Turnell-Turnef that Veronia rubicola is a distinct species. And its closest relative is Veronia bahamensis, which occurs in the Bahamian archipelago. And so with that information, again, we were able to see the Veronia lima, that third one that I mentioned, comes out sister to those two. So we have a distinct species. We know the concept we're talking about. It has a distinct biogeographic range, and its two closest relatives, which come out in the tree, we also know where they occur. Bear with me, the slide is stuck. So the next step in the genetic analysis was to look at population genetics. So I selected 10 nuclear microsatellites. These were genotyped, and I was able to observe 107 different alleles across the pool. Using this data, I was able to detect 5 separate populations, and you can see those here on the screen. Now, the interesting thing that these data were able to tell me is that the genetic diversity has been impacted over time by the reduction in the population sizes. So the island of Vieques, which has around 1% of the remaining plants that have been recorded, shows inbreeding and has the lowest amount of allelic diversity. So that is directly proportional to the population size. You can see here. So the 5 populations which we detected, you can see there's 2 on Anegada, 2 on Puerto Rico, and then 1 on Vieques. And even though the Anegada populations have over 79% of the extant individuals left, it only has just over half of the allelic diversity. So the other populations are really important for the conservation of the species and maintaining that genetic diversity. You know, so even that small population on Vieques had unique alleles to it. So conserving that population became a high priority for us. Across the historical range, as mentioned before, we found there are still plants present in all these locations. And that the reproduction of the species seems to be impacted outside of Anegada for some reason. And that was really what we wanted to get to the crux of next. So overlaid all of the modern records onto our GIS, started out with the geology. What I was able to show through this is that there's geology on each of the islands from different periods of time and history that the species only occurs on. And so on Anegada, it grows on limestone from the Pleistocene, along with overlying sand. On Vieques, the species grows on a Pliocene limestone, so 1 step backward in time. And then on the island of Puerto Rico, 1 more step back, Miocene limestone. And so what that supports is that species probably speciated on Puerto Rico and then moved eastward as those new limestone deposits were laid down over time. Species was also found in a specific type of classified habitat. And currently there's only about 90 square kilometers of that habitat, intact habitat left that the species prefers. Fortunately, there are protected areas that encompass large areas of this 90 square miles that overlies those 3 limestones. However, these protected areas only have about 1/3 of that. So there's 2/3 of the species preferred habitat that aren't under protection. Most of that is private land. And we need to work really closely with landowners to begin to develop conservation measures in those areas. So fortunately, on Vieques, all of the habitat and the geology occurs in a protected area. So that very small population is actually well protected. But on the other 2 islands, that's a different story. So very little of the habitat is currently protected on Anegada. There are proposed protected areas that we're hopeful will be enacted soon. But on Puerto Rico, around 34% of the population is currently protected. So there's still lots more to do. So as I mentioned, we were seeing that there was an issue with species reproduction. And so we worked with Fort Worth Zoo to try to figure some of this out through captive feeding, gut passage studies, and dispersal experiments with the critically endangered rock iguana, Cyclura pinguis. So this iguana used to occur across the Puerto Rican bank. Now it's only naturally found on Anegada. It's being reintroduced to a few other islands, but not within the range of Veronia. So that's, again, another big red flag. The species is dispersing. The biggest population is on the island with the iguana. The other islands, species population has declined significantly, and we're not seeing dispersal of the seeds very far away from mother plants. So this is an issue for us. We also did a lot of camera trapping, particularly on Anegada and in Puerto Rico. And we were able to find, there's many pollinators of the species. And we got some evidence, particularly on Puerto Rico, of some other birds in particular and smaller lizards that may be helping some with dispersal. But the major thing was that the rock iguana is the major disperser on Anegada. This was new. We didn't know this before. Unfortunately, one of the biggest things that came out of the camera trapping study, and again, it's great having these technologies that can be there all the time, because I had never really considered running these at night until one day I did. And then we found rats are actually taking most of the fruit. So introduced rats are probably the biggest limiter to our plant's dispersal currently. So the rats are actually eating the fruit. They don't disperse very far. They usually defecate very close to the plant, the mother plant. So we think that's one of the biggest issues is actually rat predation. So I mentioned climate change and looking at that in the history and in the future. It's in the Caribbean. Hurricanes and tsunamis have major impacts there, especially Anegada, which is very low lying. The highest point on the island is around 25 feet, 8 meters. And so one of the things I wanted to look at were the projections into the future. So I took the Intergovernmental Panel on Climate Change representative concentration pathway scenarios for 2100. Plug those into the GIS along with our digital elevation model. And began to look at how those scenarios, sea level rise, might impact the species. Fortunately, between now and 2100, up to 1 meter won't have significant direct impact on the species. But the indirect impacts of people needing to move more inland and more upslope could impact the species. But unfortunately, beyond 2100 and beyond a 1 meter sea level rise could have drastic impacts, particularly on Anegada, where we could effectively lose 90% of the species habitat if we go to the far extreme of those projections. And so that tells us that, in the longer term, if we're not able to curb the tide, literally, then we're going to have to do a lot more to conserve this species outside of Anegada to capture that genetic diversity. So through that work, and in particular, the seed banking efforts, we've accomplished a lot. We've been able to demonstrate that we can grow the species sexually, asexually. We have it secured in gene bank for future studies. We have it secured in the seed bank. We have it in ex situ collections, and it's in protected areas, and it's being monitored. So this collaboration has really paid off. Over the last 10 years, we've gone from knowing very little about the species to it now being listed in 2014 under the ESA, to there being active funds pledged toward its conservation. So a major sea change for this species in particular. I think one of the things that came out of this, though, and what I really set out to do was to try to develop a model that we could follow for other species. And so looking at that and kind of looking forward into the future, we're going to look at another example. But I think the main thing for Veronia rubicola is that we're now actually looking to use those ex situ collections and use those to reinforce wild populations and to also do inter situ. So outside, in between its native range. So material from populations in Vieques, for example, we're now thinking about planting those out in habitat on Puerto Rico, where the species has not been recorded, but there's the right substrate. We'd also want to think about, some of the offshore islands that have the right geology, but the species is no longer there. Maybe through introduction of goats in the past, these sorts of things happen. So those outplantings are really the next big step. Some of that happened toward the end of last year, led by Atlanta Botanical Garden on Vieques. And so we have plans over the next couple of years to do more of that work. Through the process, we identified lots of risks to the species in the wild. A new exotic pest attacking it, a scale insect in the top left corner. Feral animals, goats in particular, are a major problem, disturbing habitat, eliminating seedlings in the wild. And then our own impact. So the model that we use here in South Carolina of going in and bulldozing everything is the same model that gets used elsewhere. So we've got to rethink how it is we do development. But like I said, the poor seed dispersal outside of Anegada, where the iguana is, that's one of the big things that we've really got to tackle. But I think as long as this carries on, there's hope for this species. This photo was taken of a hatchling rock iguana. It had been tagged and released, and the first thing it did was went to this plant and ate that fruit. So this thing knows that's its food source. This is our connection that we've got to maintain. The group of collaborators have worked really closely. We've designed a conservation plan for the species. There's a recovery plan being published by U.S. Fish and Wildlife, which is required through the ESA. We've taken into account historic, current, and future issues, which I think is really important. A lot of plans, miss out on that, particularly the historic and the future part. But this science-based approach to conservation, this model we developed, I think that's the thing that has really had a significant impact, and we've replicated this. So the next species that I began working on is Veronia bolonis. It's endemic to Puerto Rico, same genus. Through this work, we've been able to, once again, build on the partnerships we already had, but because this species occurs in different habitats, it was also a lot of new folks involved that are, especially NGOs in the area, new forests, state forests involved in that project. So we've gathered together information about its biogeography, doing genetic research on it, and going through and working with local university to develop projects. And so that's one of the most important things out of this, is that we're helping build the next generation of conservationists in Puerto Rico that have intimate knowledge of these habitats and of these plants. We've been able to show that the species is still extant across its historic range. This was something that less than a decade ago we did not know. We thought it was left in one place. We've successfully gene banked it. We've got it in seed bank. We've grown plants out, both from seed and cuttings. All that's now being worked up into a propagation protocol. We've been able to take seed, grow them into plants, and turn them into flowering individuals. And so again, that process with a lot of species is completely unknown. It was very challenging to do. So fundamentally, this species should not go extinct. And currently we have a PhD student who's registered in University of Puerto Rico, Rio Pedras, that's undertaking a project working on this species. So by following this integrated conservation model and applying the appropriate plant conservation techniques, there's no practical reason for any extant plant species to go extinct. I'll say that again. There's no reason for any extant plant species to go extinct. We have the tools to be able to keep that from happening. What we're usually lacking is resource to make that happen, political will. So this is one of the things I'd really like for you to take away. And I hope you all will join me in trying to do what you can, either directly or indirectly, to halt this tide in the biodiversity crisis. It's going to take all of us, and even one person can make a difference. And usually what that comes down to is you making having those interactions with others. We can all do something to ensure that the future looks better for the plants and animals around us. So through my research, my experience traveling, I've developed a really strong appreciation for our reliance on plants and the importance of plant conservation. I think this statement from the Global Strategy for Plant Conservation captures this reliance very well. Without plants, there is no life. The functioning of the planet and our survival depends upon plants. There are many, many people, too many to think, here today. Staff, volunteers, interns, students that have been involved in these projects over the years. They've been supported by various funding organizations. And in particular, I have to say that without their support and assistance, none of this would have been possible. I didn't do all this. This was a network of people, dedicated people that were willing to work together. And that's the thing, and I'll keep going back to it, is the most important part of this. Thank you for your time and your attention. And if we have time, I'll take some questions.
We do have time for questions.
Right.
Yeah, you said something like there's 2,700 species of vascular plants in South Carolina. Is that right? What
Closer
percentage?
to 28. I think.
28. What percentage of those would you say a graduate of our program should? Or a faculty?
Well, I mean, this is the thing is, I've always been one that I think it's really important for you to know high level taxonomy, to know the families, to know the characters, to get down to the general level. Having a Rolodex of 2,800 species in your head, some people can do it. I think the more important thing is knowing what the resources are that you can answer the question of what is this? And I think that's the important thing of coming through a program like this is that you're learning what resources are out there, what the key characters are to look for on a plant, to know the difference between the pine and the spruce. Because at the end of the day, you may not all stay in South Carolina. I grew up in Kentucky, I wound up in London, I worked in the Caribbean, and now I'm here. And I don't know anywhere near all 2,800 plants that grow here. But my education has prepared me to understand where to look and how to go about identifying plants. I think it would be safe to say that coming out of the program, you should know hundreds of these species well, and knowing especially those keystone species in our habitats, the ones that can really help you identify what habitat you're in. I think that is really key. But I would hope that you would all focus on trying to know what those 333 priority species are, because those are the ones that I think we could make a real difference in the future.
So you said you looked at herbarium specimens. Did you collect DNA off of any of those, or was it not possible?
Yeah. So it can be challenging depending. You need to know how it was collected. So a lot of times, tropical material in particular, they don't have the ability to dry it with air drying. And so they preserve it in alcohol. And the telltale for that is that the specimen will be really black. Because the alcohol, when it's soaked in there, and then they pull it out and finish the drying process, what happens basically is all the cells explode and you can't get any recoverable DNA out. But historic specimens, I was able to get usable DNA out of 120-year-old material. And with more modern techniques than what I did. So I did the last next gen. The new next gen stuff takes even smaller fragments. And you can get a lot more genetic information out of really degraded DNA using high throughput sequencing that we have today.
You were talking about the seed viability experiment with the rock iguanas. And I'm curious on the rats, so I assume they're probably black rats, invasive black rat. Or...
The worst one on Anegada are the brown rats.
Brown rat. Did anyone look at the viability of the seeds from the rats eating the seeds? Are they destroying the actual tissue or?
On this plant, no. I did ask for student volunteers, but no one really wanted to do that. They were much more happy to process scat from the outside, coming out of an iguana, than to go around and look for rat scat. But it would be a very good thing to look at, because the iguana scat, we had 95% germination. I suspect that the seed going through the rats still has a quite high germination percentage because the seed rain. So basically, the seed the fruit that aren't being collected by an animal and dispersed, that just fall to the ground, have a very high germination percentage. But what we think is happening is that the rats are also eating, and their territory is fairly small. They're defecating in the local area. Whereas the large iguanas, they actually have a pretty big range that they cover. So we think that's limiting dispersal. But like on Puerto Rico, it's nearly impossible to find a single sapling off by itself somewhere. It's very rare. In all the surveys I've done, I've only recorded a handful of plants that you can't see an adult in the next 20 meters.
In these in situ or the like ex situ collections you have, what processes are in place to protect genetic diversity of these plants? Or is it a strong consideration?
Do you mean as long along the lines of them not crossing, or?
Yeah, because it seemed like you were talking about these cuttings, that's essentially like cloning an individual and then, not contributing to genetic diversity of plants, you're just making a clone. Is there any consideration in making sure that you're maintaining genetic diversity for the future of this population, ensuring that it has a chance to evolve over time?
Yeah. Well, the main reason for establishing that we could asexually reproduce the plants is that not all the plants are reproductive, and or you may not be able to collect fruit because there's only a few set. They don't form because of drought. That's one of the big things that we've been challenged with in the Caribbean is over the past decade in particular, they've had these severe droughts. And where these plants grow on these southern slopes, on this dry limestone with hardly any soil, it is really harsh. And so being able to reproduce them asexually means that we can capture the genetic diversity of that plant, even if we can't get seed off of it. The priority is to collect seed and maintain maternal lines of seed collections. And so those are actually separated in the seed bank by mother plant, particularly from Vieques, where there were only 6 mother plants when we started this. But being able to asexually reproduce them means that we can have plants growing in ex situ This is what we did at the end of last year. So we were growing out plants from Vieques on Mayaguez, at Mayaguez on the main island. Atlanta came in and they used the protocol that we developed at Q to take cuttings, put them through a bleach bath to clear them of any external pest disease issues, and then transport them after they've been cleaned to Vieques to propagate them, so that all that seed source material could be used for reintroduction work. So it's part of the toolbox, but yes, it's a very important consideration. We don't want to get to the point that we're just asexually reproducing one vigorous plant and we lose all the genetic diversity. And that happens. There's been studies of conifers in botanic garden collections that effectively all the trees and all the gardens were the same genotype. They've just been shared around.
Does South Carolina Botanical Garden have a next plant that they're like, you've got on your short list that you can sort of work with? Is it one of the Saracenians mentioned or something like that, or?
We're working on the list with the state botanists. So we've got a list of about 20 species that we're looking at. We're going to develop some proposals to put forward this year for funding. And from there, that's one of the, as I said before, one of the biggest issues is resource. So we've got to secure funding to help move these projects along. But what we are doing is getting involved with other projects that are going on. So we've got a collaborative project with Atlanta, looking at some magnolias. We're part of the Global Consortium for Conservation of Oaks and Magnolias, two different groups. And we're open to other places sending us genetic materials to plant out. So while we fundraise, we'll step into areas that we can handle just by having ex situ collections. But I think of that list of around 20, I think 12 of them are ESA listed species and the others are on the at-risk list.
There's a question or two online. First one from Skip. What's the effect of the non-native form on the distribution, and might the non-native wanna be filling the gold?
Very good question, Skip. At this point, the only place that we are 100% sure that the non-native iguana, so this is the green iguana, was introduced to Puerto Rican Bank. We've observed on Vieques in close association with that small population. But so far, we have never gotten green iguana on any of the camera traps around any of the plants that we've been monitoring. It could help fill that niche. If it does, we're going to have an interesting conservation conundrum to consider. But honestly, the iguana is so widespread now, I think eradication is probably not possible. They do have to do a lot of control work, especially at the airport, because it's a major concern for plane strikes when they're trying to land. But yeah, beyond that, I think potentially the green iguana could help us. My concern though is that it may be taking food from some of the native, particularly avian dispersers that we're now beginning to monitor.
And then a student, Bobby Greco, is curious what GIS you were using.
So everything that I've done has been based on Esri's ArcGIS platform. So the maps that you saw were all created using ArcMap 10. In the field, we were using ArcGIS ArcPad, which was kind of the predecessor to the current mapping projects, programs that they have for your phones. And we, in at least the last couple of trips we did, we actually used Survey 123, which is mobile device friendly.
All right, thank you.
Is that something they would?
Yeah.
Yeah. Thanks, Ray. Yeah. All right.
What is it about today?
So they like, there was a. There was I think they get out of me, but.
It's a wild land, guys. Like so many stuff. Oh my God. It's like, hey, it's like. Yeah. Oh,
Yes, I would agree.
Yeah.
So.
And then.
Next step. Very small town.
I worked at lots of homes, all the examples, they much.
I have to go
And we should.
I'm sore. I don't want to say I was like, don't need too much. Like I told them, too much first.
Yeah, well, this guy.
Yeah.
The kind of person.
Yeah,
Good friend.
Or after maybe as you get doctor.
Yeah, last we talked about, he was a professor at,
You remember being in the
I'm like,
Yeah.
Adam Alford.
But they need to like.
He's like an ag entomologist. Not just in Minnesota.
Cold. The highest must be like 70 degrees.
That's crazy. So you have a representation of like every gene. That's right. Again.
Oh.
Yeah.
Only-
I'm sorry, yeah.
Anyway.
Yeah.
Oh, yeah.
Yeah. Right now, but anyway, she now told the soil pine group.
And the veggie, the sandwich in the front or veggie.
Separate the wood, like, if you try to ID microscopically or even micro, it can't. Yeah, so that's why she now switches from the research to try to actually look at
It's amazing how fast the DNA technology.
Yeah.
So quick.
So. Like, you're good.
Yeah, she was like, sometimes, it's we get chips.
Not. Better than sell.
Yeah. I did my PhD. They finished marking starting. But like, they're taking up. Yeah, so they need to take, yeah. And also population genetics, you know, but the technique has augmented so much. So underscore like a. Oh, yeah.
Make her happy.
No, it's isolated. I'm sorry. Like around here. Yeah. I had changed a lot with me. So thank you so much for your presentation.
Thanks for coming.
Yeah, we try so hard.
I try to give away.
Yeah, I'll find people in the house studying. I don't know. So.
Yes.
Whatever that will be.
Like, it's gonna be offering.
Yeah, for the time that I'll.
I'll always offer you my cheese. Yeah. Yeah. I get it. Yes.
Yeah.
Thank you. Yeah, so.
I just want to get down to the understanding.
I really like your.
Thank you.
You just bring the chocolate.